Okay, it might be a good time to go ahead and get started. So I'm going to hand this over to Professor Ole Peterson, Editor-in-Chief of Function. Thanks.
Thank you, Chris. It's my pleasure to welcome you to this webinar, this function webinar on new insights on the cellular basis of muscle memory. We are very pleased to have Kevin Murak, the senior author of the article Nucleus Type Specific DNA Mesonomics Reveals Epigenetic Memory of Prior Adaptation in Skeletal Muscle with us today. This paper was published in the September issue of Function and also was on the front cover of the journal, as you see here. This paper has been very successful. Has been viewed more than 4,000 times already, in spite of the short time it has been in the public domain, been cited 7 times. And very importantly, it was the subject of a substantial article in the New York Times on the 5th of January 2022 in an article on how muscle memory may help us get in shape. So there's been a lot of interest in this, and for that reason, we are really delighted to have Kevin Murak with us today, who will be the speaker in this webinar. Before I give the word to Kevin, I'll just say a few words about function, because maybe not everybody knows so much about this still relatively new journal, which only started publishing in the middle of 2020. So the idea that the American Physiological Society had with function was to create a high caliber and broad spectrum open access platform for major advances in basic and translational sciences. So our aim has been consistently to publish outstanding original research findings and this is quite important, provide a broad context for these papers. So we publish a range of different article types, as I mentioned, and you may ask, well, what is then unique about function? Why did APS, who already publishes a lot of journal, suddenly decide to create yet another new journal? So we have various, I think, unique selling points. First of all, we have a fairly fast editorial process, and importantly, we make a final decision about acceptance or rejection after the first review round. And we managed to do that in, I would say, 98% of all cases. There are very few cases where we have to sit on the fence, where we will really need to see the revisions before we can decide. But in 98% of the cases, we make the decision. And of course, all articles will undergo some revision, but we commit ourselves to publishing the paper at that point in time. And this is important even in cases where additional experiments are required. We are committing ourselves to saying, yes, we want to publish your paper. Second important point is that we commission a commentary perspective from an expert in the field for every single original research article, and this provides then a broader view of the scientific impact of the article. Inevitably, not all perspectives can be published in the same issue as the original research article. Some people provide the perspectives very quickly, some people take a bit longer, but every paper will be accompanied by such a perspective. And another innovation of function is that we publish a new style of evidence reviews. So these are articles that evaluate the factual evidence in the field, providing very precise referencing to the actual discoveries by only citing original research article, rather than referring to other review articles, which then again refer to other review articles, etc. The issue that is currently in progress has several, I think, quite interesting articles. We have 2 articles uniquely in this issue on the mitochondrial ATP production, 2 important papers from the NIH, and they are also in this very issue going to be accompanied by a perspective. And we have some new evidence on the SARS-CoV-2 virus eliciting calcium signaling in epithelial cells. From the beginning of function, it was our idea that authors of papers would also be people who would speak at physical meetings, but obviously because of the pandemic, this has not worked so far. However, this year we do have a physical meeting. We have a presence, Function has a presence at EB 2022, and we have a symposium, and I have listed here the program of this symposium at EP, and we hope to see many of you at the Function Symposium in Philadelphia. And that's all I want to say at this point in time. It's now a pleasure to give the word to Kevin and hear him talk about this extremely exciting story, which has aroused so much interest. So over to you, Kevin. The floor is yours.
Sure thing. So I can share once you stop sharing. There we go. Thank you. Let's see, hopefully you all can see my screen. Can I get a thumbs up if that looks good?
Looks
Great.
good.
Awesome. All right. Well, thank you for that very kind introduction. And the title of my talk is New Insights on the Cellular Basis of Muscle Memory. And again, my name is Kevin, and I'm at the University of Arkansas as assistant professor. My lab just began in the summer of last year. And the article I'm presenting, among other articles, was published in Function last year, and I've been part of publishing in 3 articles in Function now. I've been co-author on 3, and it's been a really pleasurable experience, and I've really enjoyed the journal and the amount of attention that the articles seem to get, and they're really good at pushing out the articles and promoting them. And so I've really enjoyed the publishing process with this journal. And so so I recommend anybody that's out there listening to submit your best work there. And so without further ado, I will get into the nitty gritty here. So some of you may be interested in muscle the way that I am, but others may not be. And so I thought I'd begin just by giving an overview of skeletal muscle. It's always been captivating to me for a variety of reasons. And you know, I think people have been interested in skeletal muscle for a really long time. You can travel all around the world to different museums and see statues of ancient, really fit looking people. And this is kind of a example of a really muscular person. So this has kind of been in the mind of people for a really long time. And this is a picture of Milos of Croton, who was the most decorated wrestler of antiquity. And the story goes that he, as a young man, had a bull, and he would carry this bull with him, and did this throughout his whole life. And by the time he was fully grown, the bull was fully grown as well, and he had put on a lot of muscle mass as a consequence of carrying the bull. And this, I guess, might be one of the first examples of using progressive overload to increase muscle mass. But I say all of this just to emphasize the point that I think a lot of people do find muscle interesting for surface level reasons, and people are interested in building muscle mass for a variety of reasons. Sometimes it's aesthetic, sometimes it's otherwise. But I think muscle is a really interesting tissue to study for many reasons, not just the fact that it's, you know, visible and it can, you know, change in so many different ways and take on so many different forms. It's obviously responsible for locomotion. It contracts, it's how you get around. It has contractile material within it. It's important for thermoregulation, so shivering as an example. It's important metabolically, as it's a metabolic sink for carbohydrates, as well as an amino acid depot. It's made of proteins, so you can mobilize amino acids from those. It functions as an endocrine organ. It can secrete things called myokines, which come from the muscle that can go to other tissues and regulate the homeostasis and function throughout the body, as well as exerkines, which are things that get released during exercise that can help regulate the systemic adaptations to exercise. It's very heterogeneous. So there's a broad spectrum of different cell types within muscle. We call them fiber types. The cells in muscles are called fibers. And they may be comprised of slow twitch and fast twitch, which have very different contractile properties as well as metabolic properties. And so people could spend a whole career studying the different fiber types and how they may adapt to different stimuli, such as, for instance, exercise training or atrophy or aging. They all seem to have different proclivities under these different conditions. And so it's a really interesting tissue to study for that reason. Muscle is extremely plastic. It can grow or hypertrophy, or it can atrophy or shrink in response to different conditions. And this can happen on a pretty rapid time scale. It's a syncytium. So this is where it kind of gets a little bit, a little more scientific, I guess. But muscle is multinuclear. So the muscle fibers, like I said, are the main cells that take up the most volume in the muscle, and they're multinucleated. So which means they have many nuclei in them, which are kind of the control centers of the cell, which is in contrast to most of the cells in the body, which are mononuclear. So they just have one nucleus or one control center. And so the fact that it's a syncytium makes it really fascinating to study, but also kind of complicates things, which I'll talk about during this presentation. It also has fusogenic properties. The nuclei and muscle cannot replace themselves. And so the satellite cells, which are the primary stem cell in the muscle fiber, and the muscle can fuse to the muscle fiber and contribute new nuclei. So one cell type fusing into another isn't that common. So it's another sort of unique and interesting feature of muscle. So from an academic perspective, muscle is a very interesting tissue to study, in my opinion, and I really enjoy studying it. So I'm going to talk today about epigenetics quite a bit, and so I think it's probably prudent to begin with some definitions to help get us all on the same page so we can move forward together. For the purposes of this talk, I define epigenetics as the study of gene expression changes caused by modification of gene accessibility rather than alteration of the genetic code itself. And so DNA is organized in a very exquisite and intricate way. And the expression of genes in your DNA, so whether a gene gets turned on, so to say, can be regulated at a variety of different levels. It could be regulated at the level of the chromatin or at the level of the nucleosome via histone modifications. But gene expression can be regulated at the level of the DNA itself. The DNA is made of A's, T's, C's, and G's, and the C's can be methylated. And the methylation of those C's or cytosines can control whether or not a gene gets expressed. And this is specifically pertinent in the context of promoter methylation. So there may be a whole bunch of C's in a promoter region, whether they're methylated or not methylated can direct whether or not that gene gets expressed. And so in general, in broad terms, when you have a promoter region of a gene that's hypermethylated, you tend to get less gene expression. And conversely, if you have less methylation of a promoter region, you tend to have more gene expression. So it's one way that we can epigenetically regulate expression levels of a gene. Another term that I'll throw around a little bit is RRBS, or reduced representation bisulfite sequencing, which is the method that we use in order to measure DNA methylation. And there's another layer of regulation, which I'll talk about as well, which is microRNAs or miRNAs. So microRNAs, these little RNAs that can attach themselves to messenger RNAs and can either degrade that mRNA or block it from being translated into a protein. And so this is just like another level of epigenetic regulation, but I'll focus primarily on the methylation and microRNAs today. But one thing to keep in mind as we progress through the talk is that DNA methylation and microRNA changes in muscle are potentially long lasting. So I'm going to be talking about muscle memory. And so the fact that these things can be modified and potentially last a long time is important for that, is pertinent. So with this presentation, I aim to address these 4 questions. 1, does muscle fiber nuclear or myonuclear, that's what I'll call muscle fiber nuclei is myonuclei. Does myonuclear promoter CpG DNA methylation change with acute resistance type exercise? The second question is, does myonuclear DNA methylation change with hypertrophic exercise training? And then I'll get into what is muscle memory, which is sort of the meat of this talk. And then what is the current evidence for muscle memory? So before I go down the rabbit hole and deluge you with all this information, which I'm sure you're excited to see, is I just want to take a minute or two to call out the people that have been really integral to this research. First, I want to say that all of the experiments occurred while I was a postdoc at the University of Kentucky. I'm now an assistant professor at the University of Arkansas, but really spent the last 5 or 6 years working on a lot of this stuff with this common cast of characters, as you'll see, who I've highlighted. First, Yuen Wen, who's currently a postdoc at the University of Kentucky still and is doing brilliant work. Really excited to see where his career goes. Corey Dungan, who's been my partner in crime as a postdoc at the University of Kentucky. He's now a research assistant professor and is about to begin his own lab at Baylor this summer. And so we shared lab space for 5 years together in an office. So we're really close. Brooks Mobley and Taylor Valentino, who were working in John McCarthy's lab at the time that these studies were going on, and Taylor's actually still there. And they were really integral to the study that specifically that I was invited to talk about, which is this one, which is published in Function. Ferdinand von Walden, who was a visiting scholar in John McCarthy's lab while I was at Kentucky, and him and I just became friends, hit it off, started talking, doing science together. We still collaborate. Really awesome guy, and somebody that I spend a lot of time talking with still. And of course, I'd be remiss if I didn't mention John McCarthy, whose lab I worked in for 6 years as a postdoc and was one of my mentors. And of course, Charlotte Peterson, who's the director of the Center for Muscle Biology. And she really took me under her wing and, allowed me to do a lot of different studies as a postdoc. And I just had so much fun doing all this different work with all these different collaborators. And a lot of the ideas actually stem from my conversations when I began my postdoc with Charlotte. She kind of like handed me the football and I started running it down the field, and all these different things happen as a consequence. So I'll be try to get as much of this information in as I can. But I really just want to, pay homage to the folks that have really been integral to this work. And I just, really grateful for all my collaborators and for my mentors. So studying muscle hypertrophy, that's what I'm interested in. I've always been, I guess, a little bit of a gym rat. I like going to the gym and lifting weights, and I've always kind of had this academic interest in it as well. And during my time at Kentucky, I started spending a lot more time studying muscle growth in mice. I think it's a good model for a lot of reasons, which hopefully will become clear throughout my talk. But how do we study muscle hypertrophy in mice? Historically, we've used something called the synergist ablation approach. Mice are kind of stubborn creatures. You can't really get them to resistance train per se. And so we use a technique called the synergist ablation. And so this is a cartoon of the hind limb of the mouse. So essentially the calf muscle, this is the gastrocnemius, and underneath is the soleus, which isn't pictured. But on the medial side is the plantaris. And what we do in order to get the plantaris muscle to grow so we can study the growth process is we'll go in and we'll cut out part of the gastrocnemius muscle, so the tendon down here and the gastrocnemius muscle, and just take out that portion. And then as a consequence, when we put the mouse back in the cage and sew it up and let it walk around, the plantaris has to compensate for the lack of the gastrocnemius muscle, which is a bigger plantar flexor muscle. So the plantaris muscle then compensating becomes overloaded and will grow pretty rapidly, about 50% in a matter of weeks. So it's a very robust model for studying muscle hypertrophy, and it can considered to be a resistance exercise analog for the mouse. So here's the rub with studying muscle. It can be a little complicated because of the fact that it is a syncytium, it is multinucleated, as I mentioned before. This is an image of an individual muscle fiber. So we have a genetically modified mouse model where we could fluorescently label the muscle stem cells, the satellite cells, which reside in very close proximity to the muscle fibers. So I teased out an individual fiber with tweezers, and then we took this fluorescent image. And what you'll see is that obviously the muscle fiber is here in green. You can see the many nuclei within the muscle fiber, which are kind of the fuzzy blue dots there. On the outside, though, we have all these different cell types that are attached to the muscle fiber. So these could be anything from fibrogenic cells to endothelial cells, macrophages, but they're stuck to the muscle fiber because there's ECM around the muscle fiber where those things live, but also the satellite cells are stuck to the muscle fiber. So if I wanted to understand what was going on within the muscle fiber, it would behoove me to try and isolate out the muscle fiber nuclei, because if you just manually tease out the muscle fiber, you might get some contamination, which could then influence your interpretation of your data. So if you want to understand what's going on in the muscle fiber, it's good to try and isolate the myonuclei out. So back in 2018, John McCarthy and Charlotte Peterson's lab came up with this mouse model, which is a really great tool for the field, called the HSA GFP mouse. So I won't get into the nitty gritty exactly of how exactly this mouse works, but what's important to understand about it is the fact that we can label all of the myonuclei in the skeletal muscle of mice with green fluorescent protein. So we can basically cause all those myonuclei to light up green and fluoresce so that we can then isolate them using some different techniques. And the other important thing about this mouse model is we use this drug called doxycycline to activate the green expression, green fluorescent protein expression to label the myonuclei, but the labeling will only occur when we give the doxycycline. Once we take it away, the myonuclei will continue to remain labeled with GFP because the GFP incorporates into the structure of the DNA. However, the gene expression of the GFP will go away. And so it's interesting, we can leverage it to use it as a tool to label different nuclear populations at different points in the lifespan of the mouse. But I'll get into that a little bit more as we progress. But the important thing here is we can label myonuclei with complete control, and then we can isolate out those myonuclei for downstream analyses. And so very quickly, how we do this is we label with the HSA GFP mouse, we do a quick nuclear suspension from the muscle. So we homogenize the muscle. And then we are able to sort the fluorescing nuclei out using this laser machine called a fluorescent activated sorter. So we do fluorescent activated nuclear sorting, where we basically just shoot out the green nuclei into a tube and all the ones that aren't green into another tube. And this is a flow cytometry plot that I'm showing you here. And this shows you the double labeled nuclei, so the ones that are GFP and also have DNA, which is the PI stain. So these are the myonuclei up here, and then down here are the non-GFP labeled nuclei. And so we get these two distinct populations. And then we can take these labeled ones and we can look at them on the scope and make sure that they also have DNA in them. And basically we can use this as a way to make sure that we're getting a very, very pure population of myonuclei for downstream analyses. So it's a very, very useful tool for us. So first question, does myonuclear CpG DNA methylation change with acute exercise? So this study came about because Ferdinand von Walden, as I mentioned, was in John McCarthy's lab for a year. And he approached me one day and he asked, what do we know about DNA methylation in myonuclei during hypertrophy? And the answer to the question was, I don't think we know very much. And we had this tool and we knew how to do all the techniques. So it was, pretty easy to just say, hey, let's do a little study. Let's find out. And the study was pretty much as simple as it could be. We took our HSA GFP mice. We did the synergist ablation overload on a handful of them, and then we just did a sham surgery on the other half of the mice. And then we just compared the two. How did the overloaded myonuclei look relative to the unloaded and just the resting ones? And we were interested in DNA methylation because we think that it's a level of control, but it hasn't been studied that much in muscle. So we wanted to kind of get some preliminary unique insight into how muscle growth is being regulated at this level. And so this is again showing you that image from the previous slide. This is showing you a teased out muscle fiber. The myonuclei are green, as you can see, but then all the adherent cells are not labeled there. And this is showing you an immunohistochemical image of muscle. So it's cut in cross section, and we're able to stain it with different things. The muscle fibers are black. The muscle fiber border, which is dystrophin stain, is here in white. And you can see the labeled myonuclei that have GFP are inside of the dystrophin border, which makes them myonuclei. And then all the other nuclei are blue, so they're outside of the dystrophin border here in the interstitium and wedged between the cells. And so the labeling works really well, which we have shown many times before. So then we took our sham mice and our overloaded mice and we isolated out the myonuclei so we could do some methylation analysis downstream. But before I show you the result, I just want to call a couple things to mind. The first thing is that in the sham mice, so these are the mice that are just under resting conditions, not being overloaded, about 50 to 60% of the myonuclei were GFP positive, or 50 to 60% of the GFP signal was myonuclei, which means that the other half were not myonuclei. And I think that's a pretty important attestation to how many cells in the muscle fiber are not actually myonuclear, or are not actually nuclei residing in the muscle fiber itself. So almost half of the nuclei muscle are just things in the interstitium. And so I think that really highlights how it's important to really get in there and try to study what's going on within the muscle fiber. And then when we overloaded the mice for 72 hours or 3 days, we saw a relative shift and a reduction in the percentage of myonuclei. So this wasn't a loss of myonuclei during overload. This was instead infiltration and proliferation of cell types within the muscle fiber interstitium, or within the muscle interstitium. So all the stuff out here outside the muscle fibers. And so again, it's just important to highlight that it's important to try and isolate myonuclei if you want to understand what's going on in the muscle fiber during adaptation. And so what did we find here? We took our isolated myonuclei from the different conditions. We did RRBS, and again, we wanted to look at promoter methylation. What I'm showing you in this top figure here is CpG promoter methylation ratio. So that can also be viewed as a percentage. And what we found is that the promoter regions of genes in the mTOR pathway, as well as in autophagy related pathways, as well as the MYC gene, were hypomethylated. And so basically what this is saying to us is that processes that are related, that we know are related to growth, it seems like there's some sort of regulation at the level of DNA methylation in these genes. So that was interesting to us. And then we had some gene expression data that we could match up to this DNA methylation data and found that for a variety of the genes where there was promoter hypomethylation, we saw an upregulation of gene expression. And so I guess the take home from this is that during this brief overload period, we saw this dynamic change in DNA methylation, specifically in promoter regions of genes that we think could be important or related to growth. And so that was, I think, an important first step for us to trying to understand the regulation of gene expression during exercise adaptation. So my part one conclusions are skeletal muscle is important for whole body health. The synergist ablation approach has historically been used to study muscle adaptations to loading. To understand what is going on in muscle is informative to isolate muscle fiber nuclei or myonuclei. The HSA-GFP murine model allows for genetic labeling and isolation of a pure myonuclear population. And in response to short-term loading, myonuclear CpG DNA methylation is altered in a fashion consistent with growth processes. So the next thing is we wanted to start looking at exercise adaptation. So what happens over a longer period of time? And in order to do that, we could have overloaded the mice using synergist ablation for a long period of time, but we decided instead to take another approach. We thought that perhaps an exercise training model will give us more translatable insights to what we see in humans, since we typically don't go and cut muscles out of humans to get other muscles to grow. People just go to the gym. And so we wanted to try and replicate that. And so my colleague, Corey Dungan, was the first to put these mice on running wheels using this progressive weighted wheel running or power approach. So I'm going to show you this video here. Hopefully it'll work. But what you'll see is that there is a magnet just on one side of the wheel, weighs 1 gram, and we can stack multiple magnets on one side of the wheel because they'll stick to themselves. And what you'll notice is that the mice will run on the wheel. They do this voluntarily. Mice like running on the wheels, but they'll start running, and the way that the wheel is weighted causes them to kind of get tripped up. Like the wheel is rotating kind of an odd fashion because of the unbalanced wheel. And so they'll just run like this and stop and run. They don't always flip around, but they'll kind of start and stop all night long, kind of like they're doing sets and reps. So similar to resistance training in humans. And then we can add weight progressively over time to get them to do even more exercise. And so there's a lot of advantages here. I mean, well, the first is that you don't have to buy special wheels. You can use your current weighted regular running wheels and just add the 1 gram magnets to it. And then you can have a resisted exercise training model for your mouse. It utilizes this unbalanced wheel, which is, different from other approaches that utilize kind of a friction-based approach. And over time, the weight becomes too much for the mice to really get the wheel going. So they just will stop running. That's not the case with our model. We can gradually increase the weight over 8 weeks or any amount of time, just like with resistance training in humans. It's not really that injurious. It's definitely less injurious than cutting out muscles as we do with the synergist ablation. We can train the whole muscle hind limb. We can look at different muscles across the hind limb, soleus, gastroc, plantaris. So it's not just restricted to one muscle. It encourages continued training. So the mice will still run 10 to 12 kilometers a night on the wheels, even with 6 grams, which is, about 20% of their body weight on the wheel. So they can get a pretty robust stimulus. And the adaptations are reversible with detraining, which is really like kind of the key point for the rest of the talk. When I show you some additional data, we can just clip the wheels and then the mice will stop training, and then we can cause them to de-adapt or lose their adaptations from the original training. So that's important when you want to do a detraining study. You need to be able to stop them from training, and we can do that here. And if you're interested in ways to exercise your mice, we published a paper a couple of years ago that outlines all the new ways that people have figured out to resistance train your mouse, with power being one of them. So I encourage you to go check that out if this is the type of research you intend to do. So how effective is power? That is, I think, the first question we need to answer. We published with this model a handful of times. It began in 2019, and then we published again in 2020 using this model, and I've published at least 3 or 4 papers since then out of the Kentucky group. And so it's we've characterized it pretty thoroughly. It seems to work pretty well. But this is what we do: we take our mice that are of sufficient age. We say 4 months for the adults. We put them on the wheel without weight, and then we just exercise train them for 8 weeks, gradually adding weight to one side of the wheel until we get up to 6 grams. And then what I'm showing you here is an immunohistochemical cross-sectional image of muscle. So we take whole muscles, we cut them in half, and then cut transverse sections, and then we stain them for different fiber types. And what you can see, the first thing that's probably striking to you is this is the power train muscle over here on the right, and it's quite a bit bigger. And what you see is also a reduction in the glycolytic type fibers, which are these red ones, the 2Bs, and an increase in the more oxidative type 2A fibers. And also the muscle fibers themselves get quite a bit bigger. And so that fiber type transition is pretty consistent with what happens with exercise training, and the increase in fiber size is hypertrophy. We don't see an increase in fiber number, so it's like bona fide hypertrophy of the existing muscle fibers. And we also, in this original 2019 paper, did some additional ultrasonography work on the heart, and the heart gets bigger, it pumps better. It seems like we really generate athlete mice using this model. So it is very effective. It's voluntary. We don't have to force them. They just jump on the wheels every night. And so it's pretty low maintenance. Seems to work really well. And it was a good exercise model for us to use with our mouse model. So finally get to the question, does myonuclear DNA methylation change with exercise training? So we used our power model and we combined it with our HSA GFP mouse to label myonuclei so that we could isolate the myonuclei after training and look at the methylation profiles to see, does myonuclear methylation change with exercise training? And so this is looking at promoter regions of different genes and some pathways. And we found that this is looking at methylation on the bottom row here are the trained mice, and on the top row are the untrained mice that we compared to. And what we found is that there was promoter hypomethylation in the promoters of genes related to NF-kappa B signaling and Wnt signaling pathways. And so these are pathways that are known to be modulated by exercise training. So again, the fact that methylation changes are occurring, it's not too surprising, but it's a good confirmation that this is occurring within the myonuclei. And then we also found on the other side of the coin, hypermethylation of a variety of genes and different pathways too. First of all, we found some mild hypermethylation in genes related to PI metabolism after power training. And this is all in the plantaris muscle, I should mention. And we also found a lot of hypermethylation in the promoters of genes encoding ribosomal proteins, which was interesting to us. And we followed up on this, which I'll show you in the next slide, but this could have consequences potentially for ribosome biogenesis or perhaps ribosome specialization. Ribosomes are the construction workers of the cells, so they're important for growing muscle, we think. So we think that perhaps this methylation profile could be consequential for that. But we did a follow-up study. And I'll introduce this concept now, but I'll also touch on it in the future slides. But as a muscle grows, you tend to add more nuclei to the muscle fiber. So essentially what happens is the muscle fiber gets bigger, the cytoplasm increases, the contractile material within the muscle increases. And in order to support that growth, it's thought that more nuclei have to be added, but the nuclei can't divide themselves. Myonuclei can't, they're post-mitotic. So what happens is these muscle stem cells or satellite cells become activated, and then they fuse into the muscle fiber. So what you'll get is a bigger muscle fiber, but it has more nuclei in it as well. And the thought is that this is to support the transcriptional demands of muscle growth. And so this happens. This is a very consistent finding. We report this. Lots of people report that myonuclei increases as muscle fibers grow during resistance training. So it's a very common finding. So what we don't know is how these nuclei that fuse into the muscle fibers could differ from the myonuclei that were already there to begin with. So we had our HSA GFP mouse, and as I mentioned before, we can label the myonuclear populations at different times. So we just did a simple follow-up study where we had our HSA GFP mice, so our myonuclear labeling mice. We power trained them for 8 weeks, just like we did before. But in one group, we labeled the myonuclei at the beginning of training. So what this does is it captures the resident myonuclei, so the ones that were already there. And then the ones that fused in from the satellite cells during growth, those are not going to be labeled in this first group. In the second group, the muscle fibers grew, just like in the first group, and we labeled the myonuclei at the end of training. So we're able to capture the resident myonuclei as well as the myonuclei that fused in from satellite cells. And from these two different groups, we can infer what it is that the fused in myonuclei from the satellite cells could be contributing to the muscle fibers. So the top row here is showing you the resident myonuclei, and the bottom row is showing you the resident and the satellite cell derived myonuclei. And we're looking at DNA methylation of promoters again. And then what we found is that when you add in the fused in satellite cell derived myonuclei, there tended to be a relative hypomethylation relative to just when you looked at the regular resident myonuclei. And that was interesting to us. It suggested that perhaps the satellite cells were important for contributing these ribosomal proteins to the growing muscle fiber. And then there was a really nice data set that was already published from Dr. Charlotte Peterson and John McCarthy's lab, where they looked at the transcriptional profiles of myonuclei. So they did single myonuclear RNA sequencing with power training, and looked at the resident myonuclei versus the resident and the satellite cell derived myonuclei. They were able to do this by depleting satellite cells. And so we just went back to this data set and said, okay, well, when we add in the satellite cell derived myonuclei, are there changes in the ribosomal proteins? We would hypothesize that there would perhaps be more ribosomal protein expression, more myonuclei expressing ribosomal proteins, since it seems like there was hypomethylation of promoters in those genes in the fused in myonuclei. And that's exactly what we saw. And I picked out RPL9 because it was the most highly expressed, but it was pretty consistent with the methylation data. And so what this says to us is that potentially these satellite cells are contributing ribosomal proteins. It could again be related to ribosome biogenesis, could be important for building new ribosomes over time as the muscle grows. It could also be important for something called ribosome specialization. So you can change out the ribosomal proteins within a ribosome, and it can change what proteins or what transcripts it's preferentially going to translate. So it can change the proteome essentially. And so there's a little preliminary evidence to suggest that. And if you're interested in the concept of ribosome specialization, I encourage you to look at the work of John McCarthy. He's kind of the ribosome specialization guru in muscle. And so it's an interesting idea, and we haven't followed up on this quite yet, but it kind of got our attention and was good preliminary evidence for how satellite cells could be contributing to muscle growth after they fuse. So part 2 conclusions. Promoter CpG DNA methylation in resident myonuclei is altered by exercise training. We have hypomethylation of Wnt and FKB B pathways and hypermethylation of ribosomal proteins. The promoters of ribosomal proteins are hypomethylated specifically in satellite cell derived myonuclei. And satellite cells may preferentially contribute ribosomal proteins to growing muscle fibers upon fusion, and that could be related to ribosome biogenesis and or specialization. So let's talk a little bit about muscle memory. So what does this mean? So this idea has been in the literature for some time, and it's basically the concept that if you train before and then stop training, once you train again, your muscle will grow back more quickly. And there's anecdotal evidence for this as well as some evidence in the literature. But there are some thoughts about how this potentially happens, how this phenomenon occurs. How is it that a muscle that was trained before can recover muscle mass more quickly? And so I touched on this idea before, but I'll just reiterate it here. Let's say that you were untrained and you wanted to put on muscle mass. You go to the gym, you start resistance training. What would happen is your muscle fiber would start growing. You'd start adding new nuclei to the muscle fiber via the satellite cells. And then ultimately you get to a place where your muscle fiber is hypertrophied, it's larger, and it has more myonuclei. However, let's say you stop training for whatever reason. Let's say you got injured, or perhaps there was a worldwide pandemic, any number of things that prevented you from training the way that you would like, and your muscle fibers shrank down to the way they were before. The thought would be that the myonuclei would remain as the muscle fiber got smaller, but then as you were to start retraining, you would be able to grow back more quickly because you retained all these myonuclei. You wouldn't have to go through this process of adding in the nuclei from the satellite cells. And Professor Christian Gunderson has provided a fair bit of evidence for this in various models in mice. And so this idea has been pretty prolific in the field for some time. And so we wanted to test the hypothesis that myonuclei were permanent in our power training model. So we had this nice murine exercise training model, and we thought, okay, well, we can use this as an opportunity to test whether or not this myonuclear permanence thing is functional in our model as well. So this is what we did. So we took some C57 mice and we trained them just like we have many times before. So we had an untrained group and a power trained group by the end of 8 weeks. And then we had another group, or 2 different groups, in fact, that trained and then detrained. And so we had an age match group that we ended up combining with the untrained group. So we ultimately ended up with 3 groups. So we had the trained, the untrained, the power trained, and then a detrained group that went through the training and then just sat in the cage for 3 months. So what we saw is, which was what we expected, muscle fiber size grew with training. So this is the untrained group, this is fiber CSA, and then with detraining, muscle fiber size went back down to baseline levels. And then we looked at myonuclear numbers across these different time points, the untrained, power trained, and detrained. And we did this a couple different ways. The first way was we isolated out individual muscle fibers and counted myonuclei. We use a chemical digestion for this, so that gets rid of the vast majority of the adherent cells, so we can actually just count only the myonuclei in the muscle fibers. And we also looked at myonuclear counts using immunohistochemistry. So this is dystrophin, the muscle fiber border, and we used an automated software developed by Charlotte Peterson's laboratory to try and identify the myonuclei within the muscle fibers. And when we did this analysis in these two different ways, we found that, as expected, there was an increase in the number of myonuclei with training, but there was actually a reduction with detraining. And so this came as a surprise to me personally, because I expected the number of myonuclei to remain higher after the detraining period, but at least in our model, that wasn't necessarily what we saw. So the permanence of myonuclei may or may not be an explanation for any muscle memory of exercise training. Our results could be muscle specific or potentially could be species specific, but these results kind of got us to think a little bit differently and maybe want to look in some different areas for what could potentially be explaining a muscle memory phenomenon. So what are some other possible explanations? And the work I'm about to show you was inspired by some previous work that had been published in the human literature by Robbie Seaborn and Adam Sharples that basically suggested that there could be a DNA methylation memory within muscle tissue after exercise training that could potentially facilitate future muscle gains. So that idea kind of stuck with me personally and kind of inspired the study that we ended up doing. And so this is what we did in order to test the hypothesis of an epigenetic DNA methylation muscle memory. We used our HSA GFP mice. We similarly trained them for 8 weeks. We had a trained group and an untrained group. And then we detrained them for 12 weeks, same as before. And we had a detrained group and an untrained age-matched detrained group. And then we isolated the myonuclei, because we're using the HSA GFP mice. We isolated the myonuclei and we're able to look at DNA methylation specifically in the myonuclei. So really to try and get at this idea that there perhaps could be a methylation memory within the muscle fibers themselves. And we found a couple interesting things. First of all, we did see that so what you're looking at here, I should show you the figure first here. What we're looking at here is DNA methylation on the y-axis. And then these are the 4 groups: untrained, power trained, age match untrained, and then the detrained group. And so what we've actually found was that there seemed to be some memory site-specific CPG in the promoter region of a gene of the previous training. So this gene here, PKD201, was had a hypermethylated CPG with training. And then that same CPG was still hypermethylated after the detraining period. And there's some evidence in cardiac tissue suggests that overexpression of this gene or a knockout of this gene can result in cardiac hypertrophy. So it could perhaps be related to muscle growth, skeletal muscle growth in some way. We're not 100% sure. But we also found a methylation memory in the myonuclei of a variety of genes that are related to muscle growth in some way, shape, or form. I don't know exactly whether or not these things would lead to muscle growth if you manipulated them per se, but it was a good proof of concept to suggest that within the muscle fiber nuclei, there does seem to be this epigenetic memory, and that was in support of the previous work that was shown in humans. And so we did an additional analysis. This is from our paper from 2020, where we looked at microRNAs, and we did a very similar study design where we trained mice and then we detrained them, and we looked at the different time points. We had an untrained and a power trained group, and an age match untrained and a detrained group. And we wanted to look at microRNAs since those are epigenetic regulators as well. And what we did is we did this big analysis for microRNAs and wanted to see if there were microRNAs that would be common between the groups. And this is actually not just 3 months of training, but 6 months of detraining in this particular study. So it was a very long period of detraining for these mice, since the mice only can get to about 2 years old. So it was like a quarter of their lifespan. And we found something that was quite interesting to us. So basically, this is showing you the level of microRNA1, which is the most enriched microRNA in muscle. And it's called a myomere, or muscle-specific microRNA. And we found that with training, miR1 levels were lowered in the muscle. But then in the age-matched untrained group relative to detrained, the detrained mice also had much lower levels of miR1. So it seems like this could potentially be a memory mechanism at the microRNA level of previous training. And it's been shown that miR1 experimentally, as well as using predicted target analysis, tends to target hypertrophy-related genes. But also, if you reduce miR1 in cardiac muscle, it can cause hypertrophy. And so reduced miR1 after detraining could potentially be an epigenetic muscle memory as well, in addition to the DNA methylation that I was showing you before. So this is the final little piece to the puzzle here that I'm going to show you. So I'll try to land this plane as quickly as I can. I know I'm running out of time. So we did one final study where we wanted to retrain the mice. We wanted to look, do retrained mice that had trained previously, will they grow more quickly relative to mice that had never trained before? So that was really kind of like the big question we were trying to get at. And so we had a group where they exercise trained with power, they detrained, and then we retrained them for 4 weeks, so just a shorter period so we can kind of see if they grow faster. And then we had a group that just trained without the previous training. So it was just a 4-week training period for these mice. So they just sat throughout this whole time and then trained at the very end so we could compare them directly to the retrained group. So the white bars here are just the group that trained at the end, and the black bars are the ones that trained previously, then retrained again after detraining. Running volume was the same between these two groups during the 4-week training period here at the end. Body weight was the same, but absolute and normalized plantaris weight was bigger in the group that had trained previously. So the muscles from a whole muscle perspective seemed to get bigger faster in response to the training if they had trained before. And we saved these plantaris muscles for future molecular analyses, which hopefully I'll get to do in my own lab at some point here soon. But we had gastrocnemius muscles that we could do immunohistochemistry with. And so we found previously that the type 2A fibers, which are the more oxidative fibers of the gastroc, grew similarly as the plantaris muscle fibers do during training. So we use it as a surrogate for the plantaris. And what you see is the fiber type percentage of the gastrocs, the amount of 2A fibers is very similar between these two groups. And then the number of myonuclei between the two groups is also practically identical. But what was interesting is that the type 2A fibers in the group that trained previously grew faster than just the ones that had trained for the first time. So the control mice after 4 weeks of power. And so they were about 10% larger, in fact. So again, this is confirmatory of the fact that previously trained muscle seems to be more sensitive to retraining. And it could be related to some of the things that I've been talking about, microRNAs and potentially DNA methylation. So my overall conclusions is power is a robust training, detraining platform for mice. Promoter regions of DNA and myonuclei are modified by exercise. Some DNA methylation changes in myonuclei may persist after prolonged detraining, as well as changes in muscle-specific microRNA. Previously trained mice adapt more rapidly at the whole muscle and fiber type specific level than exercise-naive mice. So some future directions that I'm interested in is how do DNA methylation changes in myonuclei influence muscle memory and potentially enhance adaptive potential with retraining? I think the real question we need to kind of hammer out is what mediates DNA methylation status in mature muscle and what specific things are regulating DNA methylation in skeletal muscle with exercise, and what myonuclear molecular cues differentiate retained muscle from exercise naive muscle in response to exercise. So again, I saved those tissues. That's hopefully something I'll be able to work on in my own laboratory. And how do muscle specific microRNAs, specifically myomeres, control muscle mass and adult muscle with exercise? So these are all questions I'm really interested in trying to pursue as my lab gets started. So quickly, just want to, again, thank the folks that were really important for this study. This is a picture of me and Yuen Wen. He's looking very formal in his lab attire. And this was the cover of his article. I couldn't find a picture of Corey Dungan and I, even though we shared a lab for like 5 years. So it just goes to show we weren't really interested in taking pictures with one another. But this picture does capture our shared love for sour beer, though. So at least there's that. So shout out to Corey. And now that I've left, this is the dream team at the CMB that's remaining. This is Dr. Charlotte Peterson, Dr. Chris Fry, Dr. John McCarthy, Dr. S. Dupont Prestige, and Dr. Kate Kosmak. And I also want to give a quick thank you to Yvonne Fondoof-Mittendorf, who was my on-campus epigenetics guru when I was at Kentucky. She's moved on to a private research institute up in Michigan, but she's wonderful. And Dr. Julian Zerath, who's been so great to me and really providing me with a lot of opportunities, and who I still continue to work with, and who's been nominating me for all types of awards, and she's just wonderful. So I really appreciate her. And of course, thanks to my postdoc mentors, Charlotte and John. So I think with that, I can take some questions. I guess I'm supposed to be reading the questions. So let me take a gander.
Oh, thank you very much, Kevin. This is a wonderful talk. We really enjoyed it very much. And yeah, please answer those questions you have time to answer.
Great. Okay. So here's one question. Curious if you measured food consumption on the powertrain mice, they increased their food consumption to make up for the additional physical activity, or they in slight energy deficit during the power training. We have measured food consumption. They do eat more during the training. So I don't think there's I think there's a deficit. Let's see. Other questions. Boy, that one has a lot of parts to it. Let me, okay. Let's see. Interesting talk. Have you considered looking at methylation of silencer regions across myonuclei versus interstitial nuclei in your model? That's something I'm definitely interested in trying to do. Sort of what we had here was more or less a preliminary analysis, I think, and there's a lot more probably sophisticated things that we could do with respect to the epigenetic regulation of myonuclei versus interstitial nuclei. We did in our paper and function look at the interstitial nuclei, which I didn't talk about at all. And there were some genes that seem to have a memory in the interstitial nuclei as well, but we didn't go too deep into that. So I think there's a lot more work to be done here. And I think there's also, in addition to looking at methylation, we can look at things like histone modification. We can do things like attack-seek. There's a lot of additional information that we can start to glean from the data. It's just I need to get my lab started so we can do more of those things. But that's definitely things that we're trying to look into here in the future. So let me see. Let's see. Thoughts on doing a study in which MIR1 is reduced to see what impact this has on the epigenetic memory. Yeah, that's something we're actively trying to pursue right now. We're working on a mouse model where we can try and hopefully knock out microRNAs in muscle and try to get at some of those questions. Let's see. Although total myonuclei decrease with detraining, do you know if the nuclei that remain are the initial nuclei or include newer nuclei from the satellite cells? Good question. That's going to be a tough one to answer. We're going to need some more sophisticated mouse models, I think, to try and answer those questions. And so, yeah, those are definitely things that we're interested in trying to do. It's just we haven't quite gotten around to getting to it yet. Have you measured oxidative aerobic adaptations and muscle memory with the Power platform? We have yeah, we've done that in a couple different muscles. They definitely depends on the muscle you look at. Muscle fiber types are kind of weird. The type 1s are a little less oxidative than the 2As. So sometimes the oxidative metabolic adaptations don't quite match up with what you might expect. But we have looked at some of that, and they do obviously adapt. We see these massive fiber type shifts, but the metabolic adaptations aren't always exactly what you'd expect, but we haven't done like really intricate, other than like citrate synthase and SDH and things like that, the basic things. So we need to do some more advanced analyses to figure out what's going on metabolically within the muscle. Okay, are myonuclei necessary for hypertrophy? Or are they added as a coincidence of muscle hypertrophy? That is a great question. So I think, at least I'll speak for myself, my position is that I think they are necessary for hypertrophy. Ultimately, I think in order to sustain maximal muscle growth, you probably need to add new myonuclei in order to get the muscle to grow to its highest potential. I think that there is some flexibility in the muscle fiber for growing if you didn't have satellite cells, for instance. We've shown that Kentucky Center for Muscle Biology Research has been showing that for a long time, that the muscle fiber can grow some in the absence of satellite cells, but satellite cells are doing a lot of different things in the muscle niche during growth. They're communicating with other cell types, they're contributing myonuclei, doing a lot of things that are really important for the growth process. And maybe they might not be absolutely necessary to kick off the muscle growth process. The muscle fiber specifically seems to have some intrinsic capacity for some level of growth, but ultimately at the end of the day, when you don't have that myonuclear accretion, when you don't have the satellite cells doing what they're supposed to do, the muscle doesn't seem to respond quite as well, and can even become fibrotic in some instances, depending on the stimulus. So we definitely think that the myonuclei are ultimately necessary, but they're not for probably the earlier phases of growth, because the muscle fiber can do some of the work up front is what we think. Let's see, have you looked at cardiac muscle? I'm assuming with power and detraining and things of that nature. No, but I have them and it's something I want to do. I just haven't had the opportunity to get the samples out and really look at them. But something that, I'm interested in doing. I'm not a cardiac person per se, but I think it would definitely be interesting to look and see if the cardiac muscle has kind of similar epigenetic mechanisms. Would it respond more rapidly with retraining as well? And could it be related to some epigenetic component? I'm not sure yet. Let's see. Do you think this can be done in humans anytime soon? If so, how would you go about it? I'm assuming this is training, detraining, and there have been studies. The one I mentioned by Adam Sharples did a training, detraining, retraining study in humans. It was really nice. I think though, as far as if you want to isolate myonuclei and look at these different epigenetic things, I think it's on the horizon. I think the issue with doing some of the analyses that I presented today could be limited just by the availability of tissue or the ability to get at these certain cellular and nuclear populations from small muscle samples. Because in the mouse, we can collect all the tissue. In humans, you can usually just get a little sample from the leg muscle. And so yeah, I think some of these things are going to be on the horizon for human studies, but obviously we're not going to be genetically labeling myonuclei in human beings to do some of this stuff. We'll have to get clever with how we do some of these studies in order to match it up to the mouse work. Let's see. Is basal methylation different between nuclei and different fiber type fibers? I think the answer to that is yes. I think Professor Gunderson's done some work. Well, not specifically methylation, but looking at different fiber types. There was a paper published from the old laboratory where I got my PhD that showed some different fiber type differences in DNA methylation. There was a paper published last year that did it in mice that looked at DNA methylation profiles between fiber types, and it does seem like they're different. So yeah, at the point that I'm at, we're only specifying the myonuclei from different muscles, but not different fiber types. Granted, the muscles have different fiber type profiles, but I'm not going in and pulling out the myonuclei from a type 1 or a type 2 fiber, whatever the case may be. And so, but yes, it does seem like when you do look at the muscle fibers or the methylation profiles from different fiber types, you do see differences and there's differential regulation. Do you think mitochondrial DNA methylation occurs similar during the exercise? I'm not really sure. I think there was a study recently by Adam Sharples and Mike Roberts, and it was published in Facet, where they looked at some mitochondrial methylation things with resistance training. I don't remember the specific results, but there is some information on that out there. I mean, yeah, I'm not really sure within the context of my research, but there is some information out there if you're interested. Let's see. Do you think that bed rest, like a hind limb unloading model, would be consistent with your detraining training data, or do you think complete unloading may affect this pathway differently? Yeah, I think detraining and bed rest are probably two different things, because typically with bed rest, you're not loading the muscle in any fashion whatsoever, whereas with detraining, you're still walking around, you're still loading the muscles in some ways. So I would guess that they probably would be somewhat different, like different modes of atrophy, so to say. And so I would say, yeah, there probably are some differences, but I haven't looked at that personally yet. What are the potential influences of the neural component of the neuromuscular system? Yeah, we didn't look at that at all. And that's a very common question we get, because typically people think with muscle memory, it's a neuromuscular thing where it's kind of like a neuromuscular training effect where you have these movement patterns kind of ingrained, and then you can just activate them more readily. And I think it's probably an interplay of things for why we see more growth after you've trained previously. It could be related to epigenetics. It could be related to something neural. It could be related to what's going on in the interstitium, not the muscle fiber itself. It could be related to a lot of different things. So it's probably some complex interplay, and we're just looking at some little, snapshot of it. So yeah, I think that's a great question. So yeah, I think, have you looked at MIR 155? No, I have not. Yeah, still got a couple questions coming in here. Let's see. Yeah, I think I answered the majority of the questions. There was some overlap, but
in principle, we are out of time now, but if there is something very urgent that you just still feel that you would like to answer, that's possible, or some final concluding remarks if you want. But we were supposed to run for 1 hour, and we have done that exactly now. So unless you have any last minute remark, then maybe we are at the stage where we should terminate this webinar. It's been a great pleasure. It's a totally different area from my own, but I enjoyed your talk very much. It was very clear and really very, very nicely presented. So thank you very much for that. And thank you very much to our audience for being with us. I noticed that we had more than 190 people with us for the whole of your lecture. And now inevitably numbers have gone down a bit, as they always do at the end. But that's absolutely fine. So thank you very much to everybody who has been listening in. And thank you particularly to Kevin for really a brilliant talk, which has really stimulated us all very much. And thank you very much to Chris and the APS for providing the backdrop to all of this. So I think we will finish this seminar here at this point in time. But of course, it will be online in a couple of weeks' time. So there's an opportunity to revisit it at that point in time. Thank you very much.
Thank you so much. Really appreciate it. This was wonderful. Take care.
Bye-bye.